sometimes the most incredible stories are unfolding right under our noses, or, in this case, fluttering through the Canadian Rockies.
Yeah, exactly.
Today, we're diving into a really fascinating discovery in Alberta's Waterton Lakes National Park. It's about a butterfly that's, well, it's been hiding in plain sight.
That's right. For years, this little marvel was flitting around Blakiston Fan, and everyone just, assumed it was something else.
Assumed it was its close cousin, the half-moon hairstreak.
Precisely. The Satyrium semiluna. It really makes you think about how much we're still learning about, well, everything around us. The web of life,
Absolutely. And the name they've given it now, it perfectly captures the intrigue. Satyrium curiosolus.
Mm-hmm.
The curiously isolated hairstreak. Doesn't that just make you want to know more?
It really does. Curiously isolated. It begs the question.
So our deep dive today, looking at sources like a recent paper in Zookeys, is all about unpacking that. Why curiously isolated? What does this discovery tell us about evolution? And crucially, why conservation matters so much here.
Definitely. Let's get into it.
Okay, so maybe start with the basics. Why was it mistaken for so long?
Okay, so at first glance, you can sort of see why. We're talking about a pretty small butterfly.
Oh, small.
Its wingspan is maybe 1 to 1.5 inches. Delicate little thing.
Okay.
And the wings, well, the tops are this kind of subtle brown. Underneath, they're grayish brown with a scattering of black spots.
Which sounds a lot like...
Which sounds quite a bit like the half-moon hairstreak, Satyrium semiluna. So visually, the resemblance is there. For a long time, that was enough. They were just, lumped together.
But appearances can be deceiving, as they say. The real story is deeper.
Much deeper. It's in its history, its isolation.
What was the trigger? What made researchers think, hang on, maybe this isn't just another half-moon?
Well, the clue is right there in the name they eventually gave it, that curiously isolated part.
Right.
Research started to reveal that this specific group, this population in Waterton, has been separated, utterly separated from its closest relatives for an astonishingly long time.
How long are we talking?
Around 40,000 years.
Wow. 40,000 years.
Yep. Just think about that. Ice ages came and went. That kind of geographical separation for that long, it basically acts like an evolutionary incubator.
An incubator, meaning it allowed them to just sort of cook up their own unique traits.
Exactly. To develop unique genetic markers, different ecological habits.
Yeah.
They went down their own path.
40,000 years, I'm still stuck on that number. It really puts their journey into perspective. So it wasn't just like a slightly different wing pattern we missed. This long split caused fundamental changes.
Absolutely fundamental. And this is where, the really cool genetic detective work comes in.
Genomics.
Precisely. Scientists used whole genome sequencing. So mapping out the entire genetic code, the DNA blueprint of these Waterton butterflies.
And what did that blueprint show?
It showed something pretty striking. They found, and I'm quoting here, strikingly low genetic diversity within this Alberta population.
Low diversity, okay. Less variety in their genes.
Right. And alongside that, they found exceptionally high levels of historical inbreeding.
Inbreeding, okay. So low diversity and high inbreeding. Compared to who?
Compared to the nearest known populations of the half-moon hairstreak, Satyrium semiluna, over in British Columbia and Montana.
And how far away are those?
Over 400 kilometers away.
Yeah.
So quite a distance, geographically speaking.
Wow, okay. That's a significant genetic difference, then, compared to relatives not that far away, relatively speaking. Zach McDonald, one of the researchers, he pointed this out. Right?
Yeah, he did. Yeah, McDonald highlighted that this combination, the low genetic diversity plus the high historical inbreeding, it's quite unusual.
Yeah.
It's strong evidence that this little population has been doing its own thing evolutionarily for a very, very long time.
But here's the bit that seems, I don't know, counterintuitive.
What's that?
We usually hear that low genetic diversity and inbreeding are bad, like really bad, for populations' health and survival chances.
That's generally the rule, yes.
Yeah.
It often leads to problems, makes populations vulnerable.
But this one seems to have been stable for tens of thousands of years, you said.
That's the fascinating part. It does seem paradoxical. How could such a small, genetically limited population persist for so long?
Yeah, how?
Well, there isn't a definitive answer yet, but the researchers have a compelling idea. They draw an analogy with the Channel Island fox.
Oh, right. The foxes on the islands off California.
Exactly. That population also went through a period of isolation, resulting in very low genetic diversity. But they survived.
And the theory there is?
The theory is something called purging. Over many, many generations of inbreeding in this small, isolated group, harmful recessive genes, the ones that cause problems when you get two copies might have been effectively weeded out.
Ah, I see. So individuals with those bad genes just didn't reproduce as well, and eventually those variations disappeared from the gene pool.
Pretty much. It's like they went through this severe genetic bottleneck, lost a lot of variety, but maybe, just maybe, they also shed some of the really detrimental stuff in the process.
So they ended up with a smaller genetic toolkit, maybe fewer options for adapting to big changes.
Right. That's the downside.
But the tools they do have are potentially quite well suited, or at least not actively harmful, for their specific stable environment.
That's the hypothesis, yes. A sort of evolutionary trade-off, perhaps.
Fascinating. Okay, so their genes tell a unique story of isolation. But you mentioned ecology, too. Their lifestyle, their home, that's different as well.
Absolutely. It's not just the genes. If you look at where Satyrium curiosolus lives, it's quite distinct from where you typically find the half-moon hairstreak.
Oh, so?
Well, the half-moons, semiluna, they tend to like sagebrush steppe environments. Think dry, shrubby areas.
Okay.
But curiosolus, the curiously isolated one, it's found on this single alluvial fan in Waterton.
An alluvial fan that's like a fan-shaped deposit of gravel and sand left by a stream.
Exactly. And the habitat there is more like a prairie grassland. Different plants, different conditions, probably different interacting species, like ants.
And the plants are key, aren't they? Especially for the caterpillars.
Very key. The larvae of Satyrium curiosolus have a very specific diet. They rely exclusively on silvery lupine, Lupinus argenteus. That's their food plant during development.
Exclusively. Only that one plant.
As far as the research shows, yes. And what's really noteworthy is that silvery lupine isn't known as a food source for the half-moon hairstreak larvae over in BC.
Ah, so another difference. They're picky eaters, and they picked a different item from the menu compared to their relatives.
Precisely. It further emphasizes how they've carved out their own unique ecological niche, tied very specifically to that spot in Waterton and that particular plant.
So isolated geographically, genetically, and in their diet. It really paints a picture of separateness. And you mentioned ants earlier. That sounds interesting.
Yes, this is another really remarkable aspect that seems tied to their isolated evolution. The researchers documented a mutualistic relationship.
Mutualistic, like both sides benefit, a win-win.
Exactly. It's between the Satyrium curiosolus larvae, the caterpillars, and its specific ant species, Lasius ponderosae.
Okay, what's the deal they have?
Well, the caterpillars produce this sugary liquid called honeydew, basically ant candy.
Huh.
The ants love it, they collect it, they consume it, and in return,
Protection.
you got it.
Yeah.
The ants act like bodyguards, they fend off parasites, they attack predators that might go after the caterpillars. Tiny little security detail.
Paid in sugar.
That's
That's brilliant. Nature is amazing.
Isn't it? And it gets even more intricate. When the caterpillars are disturbed, or maybe if the weather gets bad, they actually retreat into the ant galleries.
Into the ants' nests, underground.
Yeah, into the tunnels and chambers where the ants live. They find shelter there.
Wow, they're practically roommates.
Almost. And get this, the adult female butterflies have been observed laying their eggs very deliberately near the entrances to these Lasius ant colonies, right at the base of the silvery lupine plants.
So they're setting their kids up from the start, laying eggs where the food is and where the bodyguards hang out.
It seems that way. It points to a really tightly interwoven, co-evolved relationship that might be unique to this specific butterfly and this specific ant in this specific place.
This whole picture is just incredible. A creature truly molded by its long solitude and its very specific little world on that alluvial fan. Okay, so bringing this all together, what does this mean for conservation? Why is recognizing it as Satyrium curiosolus, a distinct species, so important?
It's absolutely crucial because if it's just seen as a slightly odd population of the more widespread half-moon hairstreak, conservation efforts might be generic or maybe even non-existent, assuming it's secure elsewhere.
Right. But if it's its own unique thing...
then it needs its own unique, tailored conservation strategy. It faces a very specific set of vulnerabilities.
Like the low genetic diversity we talked about.
Exactly. Well, that low diversity might have, paradoxically, allowed it to persist in a stable environment for millennia by purging bad genes. It also means it likely has a much reduced capacity to adapt.
Adapt to what?
It's a change, especially rapid change, like climate change. A smaller genetic toolkit means fewer options, fewer potential solutions in the gene pool for dealing with new pressures, like different temperature regimes or changes in rainfall affecting the lupine or the ants.
That makes perfect sense. Fewer tools in the box when you suddenly need a new one.
Precisely. And this throws up some really complex challenges for conservation managers.
How so?
Well, one potential tool sometimes used for populations with low genetic diversity is called genetic rescue.
That sounds like bringing in reinforcements.
Kind of. It involves introducing individuals from related but different populations to try and boost genetic diversity, bring in new variations.
Okay, so maybe bring in some half-moon hair streaks from BC.
But that's where it gets tricky. Because Satyrium curiosulus is so genetically distinct and has been for so long, there's a real concern about something called outbreeding depression.
Outbreeding depression. What's that?
It's when you cross two populations that have been separated and adapted to different conditions for a long time. Sometimes, instead of getting healthier offspring, you get offspring that are actually less fit, less fertile, or less suited to either parental environment. Their genes just don't mix well anymore.
So trying to rescue them by mixing could actually harm them, make things worse.
It's a serious risk, yes. The genetic combinations might be disrupted, adaptations broken. It highlights how careful we have to be.
Wow.
In fact, the researchers go even further. They suggest it's possible that Satyrium curiosulus and Satyrium semoluna might not even be reproductively compatible anymore.
Meaning they couldn't even interbreed successfully if they met.
It's a possibility. After 40,000 years of separation, they might effectively be different biological species in that sense, too. If that's true, then curiosulus is truly on its own evolutionary limb.
Which means genetic rescue is completely off the table.
Pretty much. So conservation has to think differently. Maybe focus on protecting its existing habitat rigorously. Or perhaps look into establishing new, separate populations of Satyrium curiosulus in other suitable locations nearby as a kind of insurance policy against something happening to the single known site.
Like creating backups.
Essentially, yes. Spreading the risk, especially with climate change looming.
It really sounds like a delicate tightrope walk, trying to protect this unique ancient lineage that's maybe quite fragile in its genetic makeup.
It is. And this whole discovery is just such a powerful illustration of how modern tools, especially genomics, are changing the game in biology and conservation.
Julien Dupuis, another researcher on the study, he talked about this, didn't he?
Yes, Dupuis made a great point. He emphasized how genomics is really revolutionizing taxonomy. The science of classifying life and also conservation science.
I know too.
Well, traditionally, classifying species relied heavily on morphology, what they look like, physical features.
Like the wing patterns we talked about earlier.
Exactly. But this study shows that looks can be deceiving. It highlights how critical it is to integrate the genetic data, the genomic data, and also ecological data like the habitat, the food plant, the ant relationship.
To get the full picture.
To get the full picture, yes. And in doing so, we uncover what Dupuis called cryptic biodiversity.
Cryptic biodiversity, I like that term. Hidden diversity.
Exactly. Species that look very similar, maybe almost identical to a known species, but are actually genetically distinct, evolutionarily unique lineages hiding in plain sight.
And the implication is there could be a lot more of this out there.
Absolutely. Dupuis stressed that as these genomic tools become cheaper and more accessible, we're likely going to find many more examples of this hidden diversity. And that's crucial information for conservation. We can't protect what we don't know exists.
We need to know who's actually out there to plan effectively.
Precisely. We need to account for these unique, often localized lineages in our efforts to protect the whole tapestry of life.
This whole story also seems like a fantastic example of teamwork. Different kinds of organizations coming together.
Oh, definitely. This wasn't just, academics in a lab. The findings came from really strong partnerships.
Who was involved?
You had the academic scientists providing the genomics and evolutionary biology expertise. Then you had nonprofit organizations, specifically the Wilder Institute Calgary Zoo, involved. And critically, the conservation managers on the ground at Parks Canada.
So the genomics people, the field ecologists, the park managers, all pooling their knowledge.
Exactly. That combination of expertise, lab work, field work, practical conservation management, that's what produced these really significant insights. Insights that are actually actionable for protecting the species.
It feels like that kind of collaboration is essential nowadays, especially for complex conservation issues.
It's going to be increasingly vital, I think. The challenges are too big and too multifaceted for any one group to tackle alone.
And it's good to hear this isn't just a one-off discovery. The research is pointing towards next steps, future work.
That's right. This discovery opens up new questions. There's still a lot more to learn about the precise evolutionary steps Satyrium curiosulus took.
Like digging deeper into that purging hypothesis.
Maybe, yeah. and also understanding the finer details of its interactions. How exactly does the relationship with the silvery lupine work? And with the Lasius ants, how dependent are they on each other?
Lots more to uncover.
Definitely. And perhaps most importantly, there's the need for long-term monitoring. Parks Canada and the Wilder Institute Calgary Zoo are continuing to monitor this population.
Why is that so critical?
To see how it fares over time. How is it responding to environmental changes, including climate change? If conservation actions are taken, like habitat management, are they working? You need that long-term data to really know what's going on.
Right, you can't just discover it and walk away. Felix Sperling, another researcher, he had a nice quote about that monitoring aspect.
He did. Sperling pointed out how that kind of sustained, careful, long-term observation can connect all these different approaches, the genetics, the ecology, the conservation, and help answer what seem like simple questions but are actually quite profound. Questions like, why is this butterfly found only here?
It's about piecing together the whole story over time.
Exactly, building that deep understanding.
So wrapping this up then, our deep dive into the world of the curiously isolated hairstreak. It really shows that even creatures we might overlook, small butterflies in a national park, can hold just immense scientific value.
And conservation value, too. They tell us so much about evolution, about adaptation, about the intricate connections in nature.
It's a powerful reminder, isn't it, that there's still so much hidden biodiversity all around us, things we haven't recognized yet.
Definitely. Just waiting to be understood. It really underscores why scientific curiosity and those collaborations we talked about and dedicated conservation efforts, why they're all so vital, especially, in a world that's changing as fast as ours is.
It really makes you wonder, doesn't it? What other secrets are still concealed, maybe fluttering past us right now, just waiting for us to look a little closer?
A fascinating thought to end on.
Thanks for joining me on this exploration today. It's been truly eye-opening.